So hello everyone, how are you guys doing today? So hi Tasneem, hi Arvind, Kavya, Kartikeyan, Mirza, hello everyone. How are you today? Ready for another recurring session, the last one finally? When you survived the whole day, I think this should be fine. I have really tried to keep it even shorter today. So we'll just cover a few topics, okay? So Lakshmi is also here. Mirza, I think I didn't see you yesterday, so hello, welcome. I hope you saw the session, then it would be easier to follow. Good, good. So I think I will just wait for a few more seconds. Manish. Hi, Manish. Manish is here as well. Today I see a couple of new names, guys. Good. Good, good. So this will be session 2. I will start again, with the revision and those parts that were not very well clear. Again, with genetics, we have to be very patient, okay? Did you guys do homework? How many of you did the homework? And be honest, okay? I will not eat anybody if you haven't done your homework. I'm a pure vegetarian. I don't even eat eggs. So, did you do the homework? You remember you were supposed to write down with your hands the 7 characters that Mendel studied. Okay, hello guys. Can you guys now hear me? Is it okay now? I'm really sorry, this internet problem. Sometimes the internet just goes away and everything is paused like you had to, and I'm really sorry. I already took 8 minutes. I think I'm live now. Yeah, I think I'm live. Yes, I am live. I can see that. I can see myself on my phone, which is a good feeling, no? Okay, so now we were talking about homework, so we'll be very quick. Tasneem, Lakshmi, Kavya, everybody did the homework. Very good. All those who did not do the homework, just go back tonight and write down with your hands the 7 characters that Mendel studied. Which one is dominant, which one is recessive. Just write it down. You don't need to learn it and keep it on your bench where you study. Okay, good. Okay, perfect guys. So let's start. So this is the overview of the chapter, and we already know what we are going to study. We saw everything yesterday already. We discussed the introduction, important terminologies, and 7 experimental characters that Mendel did. And then in addition, we saw 2 of the Mendel's law, the law of dominance and the law of segregation. And then let's revise very quickly, okay? I went back and I did my homework, I always do my homework, and I had a feeling that some of you had little doubts in law of segregation. So I will quickly revise it. Okay, so what happens is, this is one cell here, okay? For example, this is the nucleus and there is this capital T and small t. And these are the two alleles which are two different molecular forms of the same gene. Okay, and at that time, when Mendel was doing his experiments, he did not know what genes were. He called this factors. Also, you should remember, he did not know what chromosomes were. So he just called them factors, and then he said that there was something, that was going from one generation to another. And the law of segregation says that, when both this capital T and small t are present together, they stay together as it is. And then during meiosis, they come together. There is pairing of these homologous chromosomes, which Mendel did not know, by the way. Okay. And then there is segregation, so that each of the gamete can receive either one of the allele. So this is law of segregation, that both of them will separate, both the factors will separate. Okay, the alleles do not show any blending, because that is what we discussed yesterday, that when they are present together, they do not harm each other. Their characteristics remain as it is, and that is why, if you remember, in the F2 generation, we saw 3 tall and 1 dwarf. Okay, in the F1 generation, all were capital T, small t. Okay, all were tall. But in the F2 generation, they were both tall and dwarf in the ratio, in the phenotypic ratio of 3:1. Okay, is it clear? So we just did a quick revision, because I felt that it was not very clear. And that is why when we did that question, when there is meiosis, the two gametes will separate out. They will segregate. Good? Good. Okay. And then this is the monohybrid cross. Just a quick revision, that in the F1 generation, we see all the tall plants here, and then in the F2 generation, there were 3 tall and 1 dwarf. And this is because of the law of segregation, that in the F1 generation, the capital T, small t, the alleles separated out. You see the gametes here. You see, it's capital T, small t. Capital T, small t, and this is the law of segregation, that the alleles will separate out. Okay, it should be very clear, guys. I hope now it is clear. And the next thing that was not very clear was a test cross. So what is the test cross? Test cross is crossing of offsprings with unknown dominant phenotype. Okay, one thing you have to understand, okay, Mendel did these experiments. He grew thousands of these pea plants, Pisum sativum. He grew thousands of in his garden. At that time, in 1800, when he did these experiments, he did not know how to see the genotype. Okay, he got this eureka moment because only in the F2 generation there was dwarf progeny, and then he used his brain. So to understand that when a plant is tall, if it is dominant or recessive, he did a test cross. And what is a test cross? Test cross is when you always cross it with the recessive one. Okay, always with the recessive one. So it can be either capital T, or it can be capital T, small t. And how will you see? When you cross it with this one, so for example, as we did yesterday, capital T cross with small t, what will be the gametes? It will be here capital T, small t, and all the plants will be tall. Okay, so if he does a cross of this and this, so what he will do? He will take the tall plant, which he doesn't know if it is capital T, or if it is capital T, small t, if it is homozygous tall or if it is heterozygous tall. So what he will do? He will take the tall plant, cross it with the recessive one, and if he gets all tall plants, then he knows that it is capital T. But on the other hand, now what he does, he takes this and then he crosses it with this one. Okay, so here there will be only one allele, only one type, and then he observed 50% tall and 50% dwarf. Is it clear now? So if he observes that there are 50% tall and 50% dwarf, then he can say that it is a heterozygous. So you have to tell me if both these things are clear from yesterday. Just give me a thumbs up if it is clear, guys. And then we will proceed. And I promise we have kept it very small today. So I will wait till all of you tell me that yeah, it is fine. Because see, these are the initial concepts, that you need to know, and then only we'll move to a little more difficult concepts that we'll again try to understand in a very simplistic way. Very good. Lakshmi, Mirza, Kavya. Kavya, very good. It's very clear. It's very nice to hear that all of you are understanding this now. And even now, later, if there are doubts, don't hesitate ever to ask. Okay? You are here to learn. Good. Then we proceed. So what are we going to study today? We will study the third Mendel's law. Okay, so we have studied the law of dominance, we have understood the law of dominance and law of segregation. What is law of segregation? That the two alleles, from 2n it will become n. Okay, the two alleles will go equally during gametogenesis without contaminating each other. And law of dominance, I think we all know, right? Good. Good, good. Now we come to a dihybrid cross. So what was a monohybrid cross? Monohybrid cross was for one gene. Now dihybrid cross is for two genes. Okay, now he studied inheritance of two genes. So he studied inheritance of two genes simultaneously. One is round and one is yellow. One is seed shape and one is seed color. And if you did the homework, you know that round is dominant over wrinkled and yellow is dominant over green. Okay, that is clear. So now what he took? He took these parent plants, that is the P generation here. He took round yellow and wrinkled green plants and then he formed the gametes. And just for quick revision, you remember what we discussed yesterday. How do you calculate number of gametes? 2 raised to the power of number of heterozygotes. So here the number of heterozygotes in both the cases is 0. So it will be 2 raised to the power 0, which is 1. Therefore, only one gamete is formed. Now don't get confused that there are two things in the gametes, R and Y. Why? Because there are two genes. Okay, it is still N, Parents are 2N, the gametes are N. You have to see that this is R that is 2N and here R is N. Y is 2N and here Y is N. Okay, so don't get confused with this thing. And it's very simple, If you followed the monohybrid cross, it is exactly like that, the dihybrid cross. Then you get these two gametes, capital R, capital Y, small r, small y. And when you cross them, round, yellow, exactly like the dominant parent, but the genotype is different. Okay, the phenotype is same, but the genotype is different. It is now heterozygous. Clear? Now you have to tell me what he did next. So next I will tell you what he did. He did selfing of this one. Selfing of F1. You remember in the monohybrid cross also there was selfing. There was this capital T small t and there was selfing of capital T small t. So now he did again selfing of capital R small r and capital Y small y. Now you have to tell me how many gametes will be formed from this genotype. Capital R small r, capital Y small y. So quickly tell me, how will you calculate the number of genotypes? Again, you have the formula here in front of you. I will wait. Tell me. So, in between also we will do such kinds of small revisions so that these things will be really clear in your head and you don't have to go and memorize everything, because for NIT examination you need to understand the things very well. Okay, I have answer from Kavya. I am waiting for others to answer as well. Good, Kavya. Tasneem, Lakshmi, Shivani, you want to answer? Mirza, you guys want to answer? Who else is there? Arvind? Karthikeyan, are you here today? Yeah, initially you were. Okay. Okay guys, all of you have done a good job, but the answer is wrong. We discussed it yesterday also. I know it's hectic, but try to understand, okay? So I said that the number of gametes is 2 raised to the power of number of heterozygotes. What is the number of heterozygosity here? And we did this yesterday also. It is 1 and it is 2. It is not, see, if it is capital R, capital Y, small y, then this is one gene. Okay, this is the gene that will decide if it is round or wrinkled. This is the gene that will decide if it is yellow or if it is green. Are you getting it? So, number of heterozygosity here is this is 1 and this is 2. And 2 raised to the power of 2. Yeah, see now. Yes, Manish, you were right. See, everybody wrote 16 and then Manish, yes, sorry, I didn't see it because I was explaining. Lakshmi, good. Karthikeyan, good. Okay, Kavya, is it clear? Tasneem, Lakshmi, Karthikeyan, all those who wrote 16, Mirza, Shivani, Arvind, guys, is it clear? Are you getting the point? See, don't say that if you are not getting it. I will repeat it again for you. I'll try to find even a simpler way to explain you. So you have to tell me if it is clear now. Number of heterozygosity means if the gene is heterozygous or not, okay? Because the allele itself, see, if there is capital R that is present, you cannot say if it is homozygous or if it is heterozygous until and unless another allele is present there. Because gene is formed by 2 alleles. So when we talk about heterozygosity, we talk about one gene. Good, but you have to tell me guys, all those who answered 16 before, Mirza, Kavya, Tasneem, Lakshmi, so is it clear guys? Like, are you getting the point? Because we've discussed such questions yesterday also, you know, and they will ask you, even AIMS will ask you such questions, NIT will ask you such questions. So give me a thumbs up guys if it is clear. For F1 generation, how it is 2 raised to the power of 0. So, okay, first of all, this is the F1 generation, okay. This is the parent generation, P generation. Why is it 2 raised to the power 0 is because here you see, there is no heterozygous here. Both of these are homozygous guys. Capital R, capital R, capital Y, capital Y. Both of them are homozygous, therefore it is 0. I will give you an example where heterozygosity is 1. Okay, for example, capital R, capital Y, small y. In this case, heterozygosity is 1 because see, this one is homozygous. Capital R is homozygous, capital Y, small y is heterozygous. So, it will be 2 raised to the power of 1, which is 2. So, Lakshmi, did you get it? Good. Perfect. So, guys, just keep asking, you know, if there are small questions, we can discuss simultaneously and then the doubt is clear. But remember, okay, this is the parent generation, P generation, and then this is the F1 generation. Kavya asks, can you repeat how 2 raised to the power 0? So, this is round and yellow. So, we are saying that what is raised to the power? Number of heterozygosity, number of heterozygous. There is no heterozygous here, guys. It is capital R, capital Y, capital Y. So 2 stays and raised to the power is number of heterozygosity. In the parent generation here, it is 0. 2 raised to the power 0 is 1. Therefore, only one gamete is formed. Now, if you look at this example, capital R, capital Y, small y, this is homozygous. This is heterozygous. So, therefore, 2 raised to the power 1 and 2 gametes will be formed here. Okay. And then again here, there are 2 heterozygous. So, 2 raised to the power 2, 4 gametes will be formed. Kavya, is it clear? Guys, is it clear? See, this is a little bit complicated concept. Now we discussed, right, when 2 raised to power 0, 2 raised to power 1, 2 raised to the power 2. Okay, now tell me one thing. We'll discuss one more thing. For example, now, yeah, this pen is a bit, okay, I will try. Okay, I will say it, okay. Now, capital R, small r, capital Y, small y, capital G, small g. Let's say there is another gene. Capital R, small r, capital Y, small y, capital G, small g. Here you have 3 different genes. Can you tell what is the number of gametes that will be formed here? Do you get it, guys? Like, can you tell me? Capital R, small r, capital Y, small y, capital G, small g. You have to first see that what is the heterozygosity, and heterozygosity is always for a gene. Okay, don't count the number of alleles that are present. Lakshmi, very good, very good. So you see, now I can write capital R, small r, capital Y, small y, capital G, small g. Perfect. This is amazing, guys. Thank you so much. I'm so happy now. 2 raised to the power 3, 8. Very good. Good. I think now it is clear. Yes, 8 gametes. From this, 8 gametes will be formed. Okay. Good. Let's proceed. So, from a dihybrid cross, we have 2 genes, round yellow, wrinkled green, and this is the F1 progeny. And then, this is the same diagram. See, these are the gametes. This is parent number 1, this is parent number 2. And you know this, right? This is a Punnett square. So, you have 4 different types of gametes from each parent, and they will come together. And when they come together, the phenotypic ratio is 9:3:3:1. You just have to memorize it, okay? And you don't need to memorize the genotypic ratio here because it is very complicated. There are number of genotypes that are possible. Nobody is going to ask you that. Maybe in your exam, you will be asked to draw a Punnett square, but genotypic ratio, you don't need to memorize by heart. Okay, so the phenotypic ratio is 9:3:3:1. So, how is 9:3? You see, here it is round yellow and here it is wrinkled green. And again, here it is round yellow and here it is wrinkled green. So, the dominant parent was in the ratio of 9 and the recessive parent was in the ratio of 1. And then were recombinants, round green and wrinkled yellow. You see, one gene from here, one from here. So, round, green, wrinkled, yellow. So, this was like a little bit confusing for Mendel, okay, because Mendel again, he studied like thousands of plants, out of which, some were in the ratio of 9:3:3:1. So, he was definitely expecting round yellow and wrinkled green. But what he was not expecting was round green and wrinkled yellow. So, these are the recombinants, and remember this ratio. This is a very important ratio, 9:3:3:1, the phenotypic ratio of F2 generation of a dihybrid cross. Okay, good. Then we proceed. So, on the basis of the dihybrid cross, he gave the law of independent assortment. And what does this law state? It states that inheritance of one character is always independent of inheritance of other characters within the same individual. Okay, so if we just quickly go back here to this slide, inheritance of round gene is independent of inheritance of yellow gene. Okay, if it wouldn't have been independent, he wouldn't have gotten these recombinants. So round yellow and wrinkled green are parental types. Round green and wrinkled yellow, these are the recombinants. For example, let's take a hypothetical example so that it's easy to understand. For example, there is a father and there is a mother. The father has two genes. He is tall and he has straight hair. And the mother is short and curly hair, and now the kids are born. And you know, you always have heard this, right? Whenever some relatives come to your house, or whenever if any of you has seen a baby, you know, he just looks like his father, he just looks like his mother. But when you grow up, right, it is possible, like to me, you know, people say that, you know, your nose and your hands are like your father, but your face cut is like your mother. So why is that? If there would have been no independent assortment, either the kids would be tall or straight, or they would be short and curly hair. But because there is independent assortment, the combinations tall and curly hair, straight hair and short are also possible within the kids. And the phenotypic ratio in which they are possible is 9:3:3:1. Is it clear, guys? The law of independent assortment. You see, you will see a lot of definitions, but the main concept is that if there are two genes in one parent, which let's say are dominant here. This is a hypothetical example, okay? So don't remember it. It's just for our understanding. So let's say the characters of father are dominant and the characters for mother are recessive. Okay, and then you will see, ideally, if there is no independent assortment, it will be either tall and straight hair or short or curly hair. But in the kids, you will also see that tall goes with curly hair and short with straight hair. And the parental types are always the dominant ones in the ratio of 1 and the recessive ones in the ratio, dominant ones in the ratio of 9 and recessive ones in the ratio of 1, and the recombinants are 3:3. Is it clear now? Is it clear or should I repeat? I will wait. Okay, see, that is the law of independent assortment, that inheritance of one character is independent of inheritance of other character within the same individual. Okay, there is always recombinations. There will be parental types, the dominant parental types in ratio of 9, recessive parental type in the ratio of 1. But then there will be recombinants. For example, just like Mendel observed, instead of round yellow and wrinkled green that he was expecting, he also had round green and wrinkled yellow. Perfect. Good, guys. Good. If you got it, then we can proceed. Good. So, law of independent assortment, he discovered that the combination of traits in offsprings of his crosses did not always match the combination of traits in the parental organism. And now all of you have understood it, right? That instead of round yellow and wrinkled green, the dominant one in the ratio of 9, the recessive in the ratio of 1, there were also the non-parental types. Okay, good. And another thing that you have to learn by heart is that the phenotypic ratio of a dihybrid test cross is 1:1:1:1. Very easy. But now you have to tell me, what is a dihybrid test cross? So I will tell you. So this is the F1 generation. If it is a test cross, what are you going to cross it with? Just write me down here. Write it in the chat. Be careful with the capital and small letters. I will wait, till all of you will tell me. Good. I'm happy that this concept is clear to all of you. Because, this will form the basis of what we are going to do next. So, I will wait till all of you tell me that what is a test cross, dihybrid. So, a monohybrid test cross is again with the recessive, you cross it with the recessive parent. Similarly, for dihybrid cross, you cross it with the recessive parent. So what is the recessive parent here? Tell me, guys. If you don't know, then I will help you, but just try to write it down for me. Try to be awake. Just I will just explain one more concept after this, and then we can all go any. Lakshmi, very good. Thank you. I'm very happy. See, it's very nice to feel happy. And I'm sure you're also happy after giving the right answer. Very good, Kavya. Good. Perfect, guys. I'm so happy. So you have understood now. Also remember the phenotypic ratio of this dihybrid cross that Mendel did is 1:1:1:1. Good, Tasneem. I think everybody else, I don't know if it is coming to me late or everybody else is sleeping. Don't sleep, guys. Wake up, wake up, wake up. We want to be doctors. That is why we are here. We have to work hard. This is the time when we have to study very nicely. Mirza. Okay, Mirza, I think you were not there, but see, what is a test cross? A test cross is always with the recessive parent, okay? And what is the recessive parent? This is the recessive parent. And a dihybrid cross is done to see, if it is a heterozygous or homozygous. But here you don't need to care about that. You just need to say that it is the recessive. Because the definition of a test cross is when you cross one of the parents with the homozygous recessive. Okay, one of the F1 generation, not parents, sorry, with homozygous recessive. Yeah, Lakshmi, I think they are reaching a little bit late, but okay, we will work with how it is. It's good. Good, good. Okay. I hope for Mirza it is fine now. I really hope, Mirza. Otherwise, just go through the video from yesterday again. Even still, if you have a doubt, then below this video, write in the comment section and we will help you. So, what are the 3 laws of Mendel's? Now, this is wrapping up of Mendel's laws of inheritance. The laws are law of dominance. Second law is law of segregation. Sorry for my handwriting, but I know you all know what is the answer. And the third law is law of independent assortment. Good. So these are the 3 laws. Mirza, is it clear? Like, or did you not get it? Like, just tell me if it is clear or not later, okay? Good. Then we have seen, first 4 points, and now we proceed to deviations to Mendel's law. This was very frustrating for me. You try to understand one concept so well, and suddenly the teacher says, there were deviations to this. Then you are like, oh my God. But this one is very interesting. I promise it's very interesting. So let's just build up a little bit of background. So the genetic studies of Mendel took him 8 years, from 1856 to 1863. And you have to remember it. If you guys have kept the paper next to you that I told you yesterday to write down the name of the scientist as we proceed, then you can also write down there, from 1856 to 1863, and they can ask you this question, guys. So be careful. And at that time, in that early age when not even our parents were born, nobody knew what chromosomes were. Okay, so we just called it as factors. Then what happened? Suddenly how everybody, and when Mendel was working, he was working with these plants. And he did these long studies for 8 years. And I'm a scientist. So I know whenever you do some science, whenever you make new discovery, you have to publish it in a book called journal. And then for that you have to pay money. Mendel did not have that money, so he just published it in a normal journal so that not everybody who was working with science read it. But then what happened? Then Hugo de Vries, Karl Correns and Erich von Schermach. Again, write these names, take a snapshot, write it later on the sheet of paper. They independently rediscovered the work from Mendel. A generation later, 3 decades later, they found these papers that were published by Mendel. So Mendel was already dead. Mendel was already dead when he received the Nobel Prize. He was not alive to be called as the father of genetics. He did not see that, but maybe somewhere from the top he's watching all of us. You, the future doctors, he's watching. Okay, so these were the three names who rediscovered his paper. The papers were there, they just found it and they presented it to the world. And this scientist, we have done it earlier also, that the term genetics was first used by W. Bateson. So you see on this page you have all the names of the scientists, so you have to remember that, okay. Okay, I don't know. Raj, hello Raj. So now we talk about the first deviation to Mendel's law, which is linkage and recombination. And again, T.H. Morgan, another name that you have to remember, he is called as father of experimental genetics. He discovered linkage, crossing over, sex linkage, crisscross inheritance, linkage map, mutability, a lot of names you don't need to remember now. Slowly we will see. What you need to remember is that Morgan worked with Drosophila melanogaster. And I think some of you might know, right? If you have a banana in your house and if you just keep it for like a couple of days, you know, the small flies that come and sit on that banana are Drosophila. And he studied eye color in Drosophila. So, you know, we are just building over the suspense that what is the deviation. So, we are just quickly coming on it. So, this is the dihybrid cross, right, that we have already seen here. This was what Mendel did. And what we said that the phenotypic ratio of a dihybrid cross that Mendel did is 1:1:1:1:1. So, you know, after decades that Mendel was dead, people tried to repeat his work, you know, and a lot of people repeated his work and said that it was perfect, it was great. But some people found extremely different results, and one of them were Bateson and Punnett. Write it down again, take a snapshot of this, write it down. They reported that two pairs of alleles in Lathyrus do not show independent assortment. And what is independent assortment? The third law of Mendel. Okay, that is independent assortment. So what they did, they crossed two varieties of sweet pea, one with blue flowers, one with long pollens, that are the dominant characters, with red flowers and round pollens. Now we will see what basically they did. So you see, they took blue flowers and long pollen, red flowers and round pollen. And you can see this is the dominant, this is the recessive. And then gametes are formed here. And how many types of gametes will be formed? Again, there is no heterozygous gene here. There are two genes out of which none is heterozygous. So 2 raised to the power 0, which is equal to 1. So for both, one gamete each will be formed. Okay. And what will be the F1 generation now? F1 generation will be capital B, small b, capital L, small l, which is blue flower and long pollen, the dominant character. Okay, but the genotype is different. And now he did the test cross. So you see the test cross now. What is test cross? Test cross is with recessive parent. When F1 is crossed with the recessive parent. And what did Mendel say? Mendel said that the ratio of such a dihybrid cross is 1:1:1:1:1. And what do you mean by 1:1:1:1:1? For example, he grew 1000 plants. Okay, out of those 1000 plants, 200 of them were the dominant parental types. 200 of them were the recessive parental types, and 200 each were the recombinants. Okay, so they were in the ratio of 1:1:1:1. Sorry guys, let's say he took total of 800, you know, my maths is very bad. So if he took total of 800 plants, he observed that 200, 200, 200 and 200. That is where this ratio comes from, okay, 1:1:1:1. So 200 of dominant parents, 200 of recessive parents, and then 200, 200 each of the recombinants. So this is what Mendel did. Now this is another experiment done by other scientists. When they did this test cross, they formed the gametes, they saw the progeny, And what happened now? Now the ratio is 7:1:1:1:7. Oh my god, what just happened, guys? This was like an oops moment, right? According to Mendel, it should have been, okay, just a second. Yeah. So according to Mendel, the ratio should have been 1:1:1:1. But when these people did the same experiment in a different organism, they found that it is 7:1:1:7. Why? And you know, this 7 and 7 here are the parental types. And these 1 and 1 are the recombinant types. They observed that the parental types are much more than the recombinants. And why is that? Like, this is like, oh my god, we just called Mendel the father of genetics for nothing? He was wrong? No, he was not wrong. He was absolutely correct, and these people were also absolutely correct. Okay. So then we will see why both of them were correct. So now for a second, you know, we will just see. So these are the chromosomes. This is again a pair of homologous chromosome. And then there are genes here, okay. Each of the homologous chromosome has one allele for that particular gene. And for example, this is 1, 2, 3, 4, 5, 6, 7, 8 and 9. And similarly 1 dash, 2 dash, 3 dash, 4 dash, 5 dash, 6 dash, 7 dash, 8 dash, 9 dash. So what are these 1 to 9? These are alleles, and because it is a homologous chromosome, this is one gene. 1 plus 1 dash forms one genes because alleles are different molecular forms of genes. Clear, right? So what happened? When Mendel did his experiment, see, I told you Mendel was very hard working, and that's why he was very lucky. If you work really hard, you know, you do all the questions thoroughly, and if you just leave one part, you will never get question from that part. I promise, but you have to be hard working. So what Mendel, when Mendel did his experiment, he did seed shape and seed color, and they were present on different chromosomes. And when these people did the experiments, Bennett and Punnett, they observed that the genes were on the same chromosome. So for example, just now forget about chromosomes. Just think about how do you make friends? Like, what is the easiest way to make a friend? See, when I was in my bachelor's, I did it from Delhi University. I went to the class the first day, and there was this girl called Swati sitting next to me. She was sitting very close to me. I started talking to her and we became friends. And even today we are best friends, you know. I'm here, she's there, long distance friendship. It's been 10 years. We are so close. We are still best friends. Nothing could separate us. Why? Because we were so close to each other from the beginning. Are you guys listening? I hope you are not falling asleep. Yeah, there is a lag. So I hope you're not falling asleep, because this is just the concept. Then we do questions and done for the day. So when I was sitting very close to Swati, that's why we became best friends. I did not become best friends with someone who was sitting, you know, in the other corner of the class. That is how generally you easily make friends, you know, depending if the person is nice, and she is amazing. She is my best friend even today for 10 years. So that is what happens with genes on chromosomes. How? So now you look, this is chromosome number 1 and this is chromosome number 2. And these genes are so close to each other. They are best friends, okay, just like me and Swati. Gene number 1 and gene number 2 are best friends. So what will happen during meiosis? During meiosis, these genes will try to stay as close to each other as they can be. For example, when we have tough times, you know, there are times when we cannot talk to each other for like a month or two. Those are hard times. Even in those hard times, me and Swati try to stick to each other. Whatever difficult situation we have, we stick to each other because we are so close. Similarly, the genes that are there on the chromosomes that are close, they try to stick to each other even during meiosis. Okay. So what will happen when there is meiosis? Again, there will be 4 gametes formed, but this 1 and 2, they will try to stay together. 1 dash and 2 dash will try to stay as much together as they can. There will be very little recombinants. 1 and 2 dash, this and this 1 dash and 2, they will be very less. So the closer the genes are, for example 1 and 3 will also try to stay as close to each other as they can. 1 and 4 will also try, but if you talk about 1 and 9, you know, they are very far. So okay, I mean they will try to stay close but not as close as 1 and 2. And that is why, you know, if we quickly go back here, the ratio of 7:1:1:7. Because these genes were very close to each other on chromosomes, they were seen in the ratio of 7:7. The parental types were much more than the recombinant types. Because when you are best friends, you know, you don't want more friends. Okay, so the parental types were much more than the recombinant types because they were much closer to each other on the chromosomes. For example, this is the chromosome, this was B and this was L. They were very close to each other, therefore they did show recombinants but very less recombinants. Is it clear? And you know what happened in case of Mendel? So in case of Mendel, he was very lucky because the gene for seed color and the gene for seed shape, they are present on entirely different chromosomes. So you see, if here is the color, if there is R, capital R, on an entirely different chromosome, it would be Y and capital Y. You know, it is just like you enter a hall with 1,000 people. One is on extreme right, one is on extreme left. You never would know each other, therefore you can make friends with whoever you want. That is why Mendel was lucky, you know. And this holds true for chromosomes, for genes that are on different chromosomes. Okay. So this is a very easy way to understand linkage. Is it clear? Now you have to tell me. Yeah, I think there is a lag, guys, but it's okay. We will try to work how it goes. So you have till you give me thumbs up, you know. I will wait. So the genes that are present very close on the chromosome will always stay together. They will always show more of the parental types and less of the recombinants. Perfect. And for others, just keep on replying to me so that I know that we can proceed, because this is a very important concept, guys. And if you can understand it in this way, you know, it's very nice. Good, Lakshmi. Good. Then that's perfect. Very good. So now we see how you will read it in the books. Okay, we cannot always explain this best friend concept. But what is the homework for today? That you will try to explain this concept of linkage to your parents or to your siblings in this best friend way. Just take like either tonight or tomorrow, you know, take like 2 minutes and try to explain them. It will be very funny. I used to do that. Okay, so quickly let's see what linkage, then we'll proceed to the questions. Linkage is the phenomena of certain genes staying together during inheritance through generations without any change or separation. Okay, the genes stay together because they are very close on the chromosomes. They do not separate because they are best friends with each other. And this is due to their location on the same chromosomes. So the genes that are located on different chromosomes, Mendel's law is perfect. But for the genes that are on the same chromosome, and again, you know, on the same chromosome, the number of recombinants will vary. For example, gene 1 and 2, they will show recombinants, but let's say they will show like 1% of recombinants. If you talk about gene 1 and gene 9, again they will show recombinants, but they might show, you know, like maybe 30% of recombinants, you know, more recombinants. So more the distance, more the number of recombinants, less linked are the genes, because farther the person is sitting, you are not that close friends to them. You can be friends, you can talk every day, but you will not share everything for life with them. 1 and 2 will share everything for life with them. 1 and 9, okay, we are good friends, you know. So then linked genes are exception of Mendel's law of independent assortment. So the third law of Mendel is not universally accepted and is only applied to genes that are not linked, that are on different chromosomes. Okay, and even on the same chromosome, it will depend on the distance. The closer they are, the less recombination, the more parental types they will be. Good. And then again, I am repeating this because this is important, you know. T.H. Morgan, he reported the same phenomena in Drosophila and he termed it linkage. And linkage in the genes can be identified by test cross. How? Because if the ratio is 1:1:1:1:1, then there is no linkage. If the ratio is different, you know, like 7:1:1:7 or something else, you know, depending on how close the genes are, you can identify. Linked genes refer to the genes located on the same chromosome and assort together or show very low recombination. Again, why? Because they are best friends. The genes that are located on the same chromosomes are assorted together. They always stay together. The parental types are more with very less recombination. And linkage is inversely proportional to the distance between the genes on the chromosomes. You know, for example, we just discussed this. For example, this is the chromosome, this is gene 1, this is gene 2 and this is gene 9. Between gene 1 and 2, the recombination will be very less. Because the distance is less, linkage is more. Is it clear? This distance, and because you know this inversely proportional, directly proportional can be confusing sometimes. So let's repeat it again. Linkage is inversely proportional to distance between the genes, which means more the distance, less is the linkage. Okay, more is the distance, less in the linkage and higher will be the recombinants. More the distance, you see between gene 1 and gene 9, the distance is more, therefore they are less linked. They are not best friends, but they are good friends and therefore recombinants will be more. Between 1 and 2, the distance is very less, linkage is very high, recombinants is very low. Is it clear? I hope it is clear. And now comes the part where I can relax, I can have water. And see, we are on time. Now we'll just practice some questions and then we are done. So, two genes R and Y are located very close on the chromosome linkage map of maize plants. Okay, these are two genes R and Y that are located very close on the chromosomal linkage map of maize plant. What is chromosomal linkage plan? You know, don't get confused. Do you remember I discussed this, like this is the chromosome gene 1 to 9. That is the chromosomal linkage map. When capital R, capital Y, capital Y and small r, small y, small y, genotypes are hybridized. The F2 segregation will show what? So what is the answer? Always remember, when two genes are located very close to each other, they are best friends. They will not separate out, they will not show recombination. Good Karthikeyan, good Arvind, Tasneem, Lakshmi, very good. Very good. I'm so happy that you guys understood this concept. This is one of the concepts from which there will be multiple questions. Very good. Where are the others? Manish, okay, maybe there is a lag. You guys will also answer. But I hope you got the concept. The genes R and Y are located very close to each other. Therefore, there will be higher number of parental types, very less recombinants. Mirza is saying X. I don't know what that means, Mirza. If you didn't get it, then we'll try to explain it to you later. Okay, maybe it was by mistake. Good Shivani, good Mirza, very good guys. Good. Next question. Okay, not the next question, just a quick revision. So in this one, we repeated law of segregation. We discussed about test cross. We discussed about the law of independent assortment, the third law. And then we saw deviation to the third law, which is by linkage, that when the genes are present very close to each other, they are best friends, they do not separate, we'll see more parental types and very less recombinants. So let's revise. We'll just do a couple of questions, not a lot, very few questions, and then you can go and eat and relax. So the next question is, select the correct statement from the ones given below with respect to the dihybrid cross. And you see, these are all the questions that are previously asked. I did not make any of these questions. So, these will be a bit confusing. Read them properly. I will not give any hint in this one. Okay, I will see what you guys answer and then we will see how it is. And then I will drink water. Everybody has to answer. See, I'm so happy, guys. You really understood the concept. Thanks a lot. You were paying attention. You are really good students. Nobody can stop you. Keep working hard just like this. Good, good, Kavya, Kartikeyan, Arvind, Lakshmi, Tasneem. Very good. Very good, Mirza. Very good, Shivani. Amazing, guys. I'm so happy. Good. Let's proceed. So the right answer is D. Next question. So this is something, in a test cross involving F1 hybrid flies, more parental type offsprings were produced than the recombinant types. What does this indicate? More parental types than the recombinants. So you always have to remember, in your mind the best friend concepts. The ones that are close, nothing can separate them. They are linked. So more parental types. Good, very good. See, you can solve now so many questions easily. Very good, Shivani, Kavya, Lakshmi, Kartikeyan, Tasneem. Very good. Good, Arvind. Very good. Good. So then let's proceed. Okay, everybody of you understood it. Everybody I see is giving the right answer. Very good, Manish. Very nice. Next question. Pure tall pea plant can be differentiated from hybrid tall pea plant by. And what do you mean by pure tall and pure hybrid? They are basically asking you how you can differentiate between capital T and capital T small t. This is a tricky question. I have given you a hint, So how can you differentiate them? Try to give the answer for this. We have discussed test cross yesterday also and we have discussed this today also. And I think this is almost the last question and I think we have just one more question after this. Then I will give you again a very little homework. Kartikeyan, very good. Kavya, very good. Who else wants to write? Yeah, I know there is a lag, but okay, we will wait. Tasneem, good. Okay, I will start explaining. Everybody answered it right in their own way. Okay, see, what we have studied is test cross, right? Test cross is used to find the progeny. But here, they are talking about a monohybrid cross. Obviously, if there is a test cross, then you can find it. That is what we studied directly. The second is selfing and finding the progeny of all tall plants. That is also possible. Why? For example, if this is the genotype of the F1, you can self it. If you get all tall, then you know that it is dominant. Okay, the second thing you can do is, you can again self this one, the heterozygous one, and then you will get the normal Mendelian ratio. You see, if you get the normal phenotypic 3:1 ratio or genotypic 1:2:1 ratio, you know it was a heterozygote. But if it is a homozygote, you will just get all tall plants. So that is another way. Therefore, the answer is 4. See, this is how, confusing questions will come in exams. So there are some things that we read by book. Test cross is used to see if it is a homozygous tall or heterozygous tall. But we also did this selfing, right? You remember, in a mono hybrid cross. So you have to, read through the options very carefully and use a little bit of your, knowledge to find out. That is why, yesterday also I said, it is very important when you practice such kinds of questions. So, now next time if you will see a similar question, you know what to do. Okay. See, C is not the answer because they are saying you self it and then you test cross. Okay, so what they are saying is that first you self it, like you self capital T crossed with capital T, then you get capital T, and then you do a test cross. So they are asking you to do 2 steps, which is not what you will do. Why you want to increase the time? Is it clear? And again for capital T, small t, you have to, do a test cross again with small t, small t. Everything you have to cross with small t, small t. So that is what it means, selfing and test crossing. They don't mean that either you self or you test cross. Otherwise, they would say, either you self or you test cross. They will be very simple with the language of the question. So good that you asked this question, Kavya. That is why it is not C, because selfing and test crossing. So they mean first you will self them and then you will do a test cross, which is not the case. You don't want to do more work when you have simple options to do. Okay, good. And then, so the answer is 4D. Okay, try to, go through questions. I'm always saying that. And if you have any doubts, we are there to help. You can write in the comment section. You can write us an email. We are always there to help you. This is the last question, which is an AIIMS question. AIIMS Delhi has a very beautiful campus. I did my master's from AIIMS Delhi. And I tell you, it is such an amazing place to be. And you all can be there if you do hard work. So this will be, AIIMS question. AIIMS will scare you by giving you these big questions and everything. But if you know the concept, if you especially know the best friend concept, this question you will just answer like this. Okay, so let's read. The experiment shown. Don't look at these crosses right now. Let's just read the question first. The experiment shown in the figure has been carried out by Morgan. Okay, the Drosophila, to show the phenomena of linkage and recombination. If in cross 1 genes are tightly linked, they are best friends, and in cross 2 genes are loosely linked, they are friends but not best friends, like good friends, then what will be the percentage of recombinants produced in cross 1 and cross 2 respectively? You see, here is the cross 1 and these are the 2 chromosomes, and these are Y and W gene that are located very close to each other. Okay, these are the 2 X chromosomes, this is X and Y chromosome, and this is the F1 generation here. And again similarly for cross 2, so the main thing that you have to just see here, that here the genes are very close, here the genes are little bit apart, and then you look at the options and you tell me what is the answer. Okay. So good. Kartikeyan, Tasneem, Kavya, Arvind, very good. I am waiting till I get the answers here, because there is a lag. Let's just wait. It's anyways the last question, then we can go. Anybody else wants to reply? Manish? Who else is there? Karthikeyan has replied. Tasneem Mirza. Okay, good. So everybody of you replied B. You see, you can see the AIMS question. You can answer it. Good. Kavya said maybe B or D. Okay, Kavya. So for everybody, this is very important because there can be this confusion, you know. Obviously, like, see, we know that for the cross 2, the number of recombinants are going to be more than cross 1. And this is the thing and this is the thing there. So there is confusion between these two. But you know, 62.8% is a very high percent of recombinants for the linked genes. Okay, it is very high. It is not that high. That is why the right answer is 1.3. So whenever you have such options, just look for the one that has the lowest one. Okay, because when you have linked genes, when they are best friends, 62.8% is very, very high percentage. Is it clear, Kavya? Kavya, is it clear? For everybody else also, you know, sometimes you can have this kind of confusion. So I will wait, you know, till you guys tell me that, yeah, that is clear, because 62.8% is a very high recombination frequency. Okay, 1.3 is good. So whenever you have such two options, you look, because you see, they are so close to each other, you know, why won't they be best friends? Obviously, they are best friends here. Good, Lakshmi, good, Kavya, good. Okay, and that's it for today. And now is my time to ask you something, is for the homework. Okay, so what is the homework here? Read page number 84 of your NCERT. You don't have to read, it is just one figure you have to go through, and tomorrow I will ask you if you understood that or not. It is a surprise. You can, if somebody of you is very curious, you can go before dinner and look what is at page number 84. Okay, and also, you know, again I am telling those who of you did not do the homework yesterday, just write all the 7 Mendel factors. Write it with your hands. Please don't do printouts. And also write the name of all the scientists. Okay. Okay guys, I hope it was all clear today. I really hope, because this is a very important concept. And see, as I promised, I tried to keep it a bit small. I am 10 minutes ahead. But okay, we'll try to keep it even smaller yesterday and more interesting. Okay, so all of you can get it. So good night, good evening. Have a very nice dinner. Work hard, guys. See, there are recurring sessions, there are recurring classes. You have to do school syllabus, then you have to prepare for NEET. But that's what you signed up for, right? You want to be a doctor and nothing can stop you. You see how you can solve in every lecture AIMS questions and NEET questions. You know, all these questions I'm taking from NEET. So you can do it, okay? Just keep on the hard work very high. Good then. Have a nice evening. Good night. And if there are questions, just keep on writing in the comment section. We'll try to help you. Good, Shivani. I'm very happy that the concepts were clear. And you can now enjoy your food, but please read page number 84 of NCERT. It's just one figure, and I think if you understood the concept today, you will get it very clear. Okay, now I'm going to log off and see you guys tomorrow. Same time, same place. Hopefully my internet will work fine and everything. So, thanks for being patient, you know. In the beginning also we lost a couple of minutes, you know, because of the technical errors. Okay guys, good night. Have a nice dinner. Bye.